Good afternoon. This is Dr. Dan Guerra from VerEvMed Studios in the Pacific/Inland Northwest of the United States. Today we're going to get into a couple of research papers that are going to be examining a common antimicrobial in the environment that's used in various human products and whether or not this household product may actually impact human disease. So I'm going to be looking at one important primary paper that just came out in mid-2018, and then I'm going to back that up with several other papers that I've read and that we're going to use data from to examine this complicated discussion. So let's get started. Again, that's me, Dr. Dan Guerra. I am the co-founder and chief scientific officer of VerEvMed. VerEvMed's email is right there. Our website is there. Please contact us if you'd like to be one of our clients. What we do is get into the scientific literature for you and examine anything that you might be interested in. And you can come from various levels of education and walks of life, from people who have high school education or high school students to college students, people who have graduated from university and have jobs, that are interested in something about biomedicine and want to know what the science says behind it, to physicians, to veterinarians, to lawyers, and to any other professionals that have advanced degrees. And the same kind of thing, basically to look at the scientific literature. We don't mess around with anything that's published in newspapers or anything that hasn't gone through rigorous review via peer review assimilation, which is normal for scientific papers. Of course, there's a huge caveat there. Just because something is published doesn't mean that it is bona fide. So that's why I call it VerEvMed. We verify the evidence in the research, comparing it foundationally and coherently with the other research, and also using logic and other foundational parameters we can find in primary reasoning to determine finally what the literature may say on a given topic. So that's what we do at VerEvMed. Okay, there I am again, Dr. Dan Guerra. I have a PhD, not an MD, and I'm a biochemist, a lipid biochemist. That is my specific email there, djgphd@gmail.com. You can contact me on any of the content of any of these YouTube VerEvMed video lectures that I've set forward in the last year or so, year and a half. Again, I'm trying to put these all up on my website. It's been a slow process because I don't have any help. But I do have all these on my Facebook page, which is right there. And so you go to my Facebook page, you're going to find all of these videos. Please contact me directly at my email if you'd like to discuss this or if you'd like to get involved in VerEvMed, and we'd be glad to work with you. Okay, so let's just get started. Slideshow from the beginning. Okay. We're going to discuss Triclosan-mediated inflammatory bowel disease, that's furthermore called IBD, and the advancement potentially to colorectal cancer. Does a common household antimicrobial play a role in the incidence, severity, and spread of IBD and complications leading to chronic disease? It's a question we're going to try to answer today by looking at the literature. All right, so let me close this here. We don't need that. There we go. Okay, let's move on. Okay, as an introduction to the paper, this structure here is actually triclosan. It is a diphenyl ether, okay? You can see that there. There's the ether linkage and there's the two phenyl groups. Also notice that's an hydroxyl group. That's going to be really important later when we talk about mode of action. Triclosan, or we can abbreviate it TCS as they do in the paper. By the way, the paper is published in Science Translational Medicine, an excellent journal. I suggest anyone that's interested in taking a look at how science is directly impacting medicine. This is a good journal to take a look at. I believe it is under a paywall, so you have to have a library access to it. Paper was published just a few weeks ago, May 30th of 2018. Today, by the way, is June 9th, 2018. So not even 2 weeks ago. Okay, so TCS is a diphenyl ether. It is also a fatty acid synthase type 2 eno-reductase enzyme antibiotic inhibitor. So you have two different types of fatty acid synthase, or FAS. Type 1, which is what humans have and yeast has, eukaryotes in general, and type 2, which is found in microbiota and interestingly, in higher plants. Now, the only distinction you need to know about type 1, type 2 FAS is that type 2 involves soluble discrete proteins as each of the enzymatic reactions, whereas type 1, which has variable subisoforms, is a polyprotein. And therefore, antibiotics that target type 2 fatty acid synthase can be arguably good antimicrobials because it has a different structure. A different protein structure means that inhibitors, pharmaceuticals in general, are going to attack that protein complex differently in terms of the pharmacodynamics. And as such, you can generate compounds that might target just the type 2, even though the reactions are identical. Because of the structure of the protein, remember biochemistry is all about structure function. Because of the structure of the polypeptide being soluble discrete proteins, a compound like triclosan can get into an active site of an individual protein, which might be otherwise occluded in a polyprotein, such as FAS type 1, like in humans. And therefore, you don't affect, at least theoretically, the host fatty acid synthase system, which would be, of course, deadly, and only attack the microbial one. But as we'll see, that is using the belief and supposed knowledge that the compound you're looking at only has one target. That is, it only has one mode of action. There is nothing I've ever seen in any pharmaceutical, any compound, any structure that has carbon in it, even things that only have metals, that only affect one individual, let's say, catalyst, like an enzyme protein complex. So that means there's always off-label effects, and we're going to see this big time with triclosan. All right. Anyway, it is, anyways, out there as being a type 2 enoreductase enzyme inhibitor. You find it as an antimicrobial additive in thousands of different types of consumer products, including hand soaps, the kinds of things you see at the sink, toothpastes, cosmetics of various kinds. It's also painted onto clothes, kitchenware, and indeed children's toys. So triclosan is ubiquitous. Because of that high volume use of the compound, the entire population is actually exposed to it at every stage of life, from even in utero to early post-parturition, all the way through early childhood, adolescence, of course, and puberty, those very difficult periods of time for humans, as well as young adulthood, middle age, and the elderly. Everyone is impacted by TCS in the Western world and increasingly worldwide everywhere, including developing countries. According to the National Institutes of Health Examination Survey, they say about 75% occurrences tested in U.S. urine analyses. So that's like, what, 3/4 of all urine that's been tested in the U.S. Finds triclosan in the urine. So pretty ubiquitous compound. The regulation of TCS used in consumer products is relatively unregulated, but recently, September of 2016, the FDA issued its very first regulation, thereby removing TCS from certain over-the-counter hand washing reagents. Since soaps containing TCS, now get this is why they said they were pulling it, demonstrated no consumer health benefit. Now, of course, when you see that, that's a negative, right? But you also know that possibly TCS could be doing something different. The biggest thing you can think about here, before we even get to the paper, is antibiotic resistance. Once you use an antimicrobial or an antibiotic, what happens is you develop resistance to that compound by bacteria that develop genetically resistance alternatives. And there's no difference here. In fact, there is a enoreductase that bacteria have that completely obviate the mode of action of triclosan and are therefore completely active in its presence. This has been published extensively in fatty acid synthesis, microbial bacterial fatty acid synthesis literature going way back to the early 90s. So we know a lot about mutants for this enzyme that are completely resistant to it. So that's out there. So we already know that there is an evolutionary patterning of directed towards being immune to triclosan. Okay, so that's not mentioned in that report, but that's obviously the positive side of all this. So anyways, the effects of TCS on colonic inflammation and associated colon tumorigenesis, which are on the rise worldwide, particularly in the Western world. Anyways, TCS on that disease is unknown. So these folks did this study and basically wanted to see whether that TCS had an effect in colonic disease. And they're using a murine model. All right. Sorry for the slide, but it was hand done years ago, so I like it. All right. So it's just showing you all the structures of fatty acid synthase. Okay. This is acetyl-CoA, right? Make malonyl-CoA. You do a condensation reaction after you first make a thioester with acyl carrier protein. So acetyl-ACP, malonyl-ACP, condensing now to the C4 intermediate. Then you carry out a reduction, a dehydration, a second reduction, utilizing either NADPH or NADH. This is the triclosan target here. This is the enoyl reductase, ene because there's a double bond there. See that transient trans double bond, and enoyl reductase, going to add hydrogen on that bond, and you're going to end up with the next saturated series of the fatty acid. And then you just continue the fatty acid synthase reactions, adding malonyl ACP, driving that reaction by releasing carbon dioxide, shown here, and then making it up to C6 to C16. Palmitic acid is the most common major end product of de novo FAS. So these are soluble discrete proteins in, again, FAS type 2, not in FAS type 1, where these reactions all exist but embedded within a polyprotein. All right. So here's more detail about it. Here's fatty acid synthesis reactions. There's, the first one was the ketoacyl synthase. Remember I told you, taking any acyl ACP, adding the enzyme itself, making acyl beta ketoacyl ACP synthase and free ACP. Ultimately, you're going to make the next, the ketoacyl product associated with ACP once you move that carbon off of the enzyme surface. The next reaction is the beta ketoacyl reductase, or KR. You're now taking that keto function, you're making an alcohol, you make the beta hydroxyl. You get the water removed across that bond, right? Yeah, you make the enoyl ACP, a variable chain length. Of course, water is the product. And then here's your enoyl reductase. And in bacteria, it's primarily NADH is the reductant. And that's, again, the target of triclosan. Here it is one more time. You have this trans 2 enoyl ACP. It's a trans double bond. You see there. No scary stuff about trans fatty acids, by the way. Totally rubbish that there's something wrong with fatty acids in the environment that are of the trans nature because they're common and ubiquitous. You metabolize them just like this. Anyways, trans 2 ACP then gets reduced to acyl ACP, fully reduced form. And there again is a nice version of triclosan, which give you a little bit more than just calling it a diphenyl. It's 2,4,4' trichloryl 2-hydroxyphenyl ether. Okay, see these chloride ions here. It makes it quite soluble. But this hydroxyl group here is really considered part of the major mode of action, as again I alluded to earlier, and we will see later. Now, not only do fatty acids get through de novo FAS, they also get elongated and desaturated particularly in human systems and in eukaryotes. So once you make palmitic and stearic acid, you then go through a series of desaturations, but you also go through a series of elongations. And the elongations, see here, are basically embedded FAS enzymatic pathways that make the C from the C16 to the C18, C20, C22, all the way up to, here's docosahexaenoic acid, right? 22 carbons with 6 double bonds, okay? Or icosapentaenoic acid, 20 carbons with 5 double bonds. So these are all metabolic end products of fatty acids taken in the diet. Now I'm bringing this up because the elongation reactions in humans are not the same as the de novo FAS. Elongation reactions tend to be associated more with the mitochondria or the peroxisome or the endoplasmic reticulum. And these enzymes are indeed not of the FAS type 1, which means they could be targeted by triclosan, which for some reason is never really discussed when we talk about triclosan being used as strictly a microbial, microbiocide or microbial, antimicrobial. I want to bring that up because those could be potential targets, which are not mentioned in these papers, but which I bring up every time I teach this in lipid biochem classes. All right, here's the paper, Science Translational Medicine, 30th May 2018, volume 10, etc. What do they say? They say common mouse diet. This is now how the work is done. They take a common mouse chow and they supplement with either 10 or 80 parts per million TCS. They do it for 3 weeks. They feed the mice 3 weeks on this diet. And then they do an analysis of their plasma and its conjugate which is called TCS glucuronate, which is basically the first way you degrade the TCS. And they're using liquid chromatography mass spec tandem. The results of the plasma levels in the mouse model were a good fit with concentrations of the same triclosan compound you find in human volunteer in the urine. As I said, also you can find some of this in sweat or any mucous membrane secretion. So using 10 and 80 parts per million was not arbitrary and capricious on the part of these people doing this work. It actually results in concentrations in the sera, or in the plasma here, of this murine model, which can be phenocopied and mimicking what happens in humans just living out in the environment. All right, first set of data. Exposure to TCS increases basal inflammation in mice. Mice treated with 80 parts per million TCS had increased spleen weight. Okay, so here's 10 parts per million, 80. You see an increase in spleen weight. This means you're getting an immune response. An increase in IL-6, that's a pro-inflammatory cytokine in the plasma. Okay, you see there's an increase. And the 80 is actually different from the control, and so is the 10 parts per million. Also, the expression at the transcriptional level of interleukin 6 is increased, okay? So this is all happening in the colon, right? You also get some histopathological disorders because of the TCS. Here's the control. There are the crypts within the colon of the mouse. You see when you add some TCS, you start to get this disruption of the structure. And by 80, you get a complete dissolution of the microvilli of the gut. So this is illustrative of enhanced systemic and colonic inflammation. And this is given the histological score here, showing increased inflammation of the colon. And you can see here, as you increase the amount of TCS, you get more. Okay? Statistically different. So it looks like TCS does cause inflammation and does increase plasma levels of circulating interleukin 6, which is a pro-inflammatory cytokine. Continuing. Exposure to TCS increases the dextran sodium sulfate-induced colitis. So now they're adding dextran sodium sulfate. This is showing you how the experiment's done. The diet contains TCS for all 3 of the weeks, right? But the last week, they're giving them this dextran sodium sulfate, and that induces colitis in the mouse model. So take a look at this. Here's the control, and here's the TCS. You can see that you get a decrease in colon length. That's what this is showing you here. This is mouse colon here, not humans, don't worry. And so there's a decrease, which means it's being destroyed, or something's happening to the colon where it's not having its normal projected growth. Here, and again, you can see the severe damage using TCS between the control, and you're looking out at this histochemical staining of using 80 parts per million, and here is the score itself. Now, this is when you're inducing colitis. So when you induce colitis with this dextran sodium sulfate in the presence of TCS in the diet, you get even a worse destruction of the colon, right? Lots of inflammation. Also, you get the increase in 3 different cells, innate immune cells. You get an increase in total leukocytes, which is shown you here, an increase in macrophages, circulating macrophages. These are just the surface markers that tell you that's a macrophage, and this is a neutrophil. Okay, these are just the proteins on the surface of these cells telling you that this is a neutrophil, this is a macrophage, and that's just general leukocytes. You see, as you increase, as you increase the amount of triclosan, here's just showing you between control and 80, you get a statistically increase of leukocytes in the blood, macrophages in the blood, and neutrophils. Likewise, you're still getting this IL-6 in the plasma junked up, right? And also you're getting the actual gene expression in the colon increase. So you're showing you it's a transcriptional event. All right? If you add additional TCS to the diet, now this is like saying, okay, now that we've got this dextran sodium sulfate-induced colitis, and we've been feeding the animals this 10 or 80 parts per million TCS, now let's add a little bit more to the diet. Let's see if we can exacerbate it. And you see that there is worsening between the control, 5 parts per million, and 10 parts per million. So you get an increase in tissue damage. So that means that any other increase in TCS is going to further cause colon damage and inflammation. Figure 3 of the paper shows you exposure to TCS increased colitis in interleukin 10 ablated mice. So this is a knockout mouse where you don't make interleukin 10. What is interleukin 10? It's a cytokine, but it's an anti-inflammatory cytokine. So you have pro-inflammatory, anti-inflammatory cytokines. IL-10 is anti. Okay, so The way they did the experiment, once again, the diet always has TCS, and then they add this piroxicam at week 12. Now, what's piroxicam? It's a steroidal anti-inflammatory. Okay, so they're just adding that to see whether or not they can impact the inflammation. Once again, you're looking at leukocytes being increased, okay, even in the presence of this antisteroid, this steroidal anti-inflammatory, and you get an increase in leukocytes, you're getting an increase in macrophages. Again, those are their molecular signatures on the surface. You also get an increased expression of interleukin 6, even in the presence of the anti-inflammatory, and you still get this terrible damage to the colon, showing you this histological score just looking at the staining of that tissue. Exposure to TCS increased the dextrose sulfate-induced colitis in mice. So do you cause disease? And does this enhance it? Oh, sorry, that went the wrong way. Exposure to TCS increased the azoxymethane DSS-induced colon tumorigenesis in mice. So now you're adding one more ingredient. You're adding this azoxymethane, and azoxymethane, actually shown here when it was delivered, actually induces colon tumorigenesis. Okay, that's what this compound does. Okay, it's an agent of tumorigenesis. You can see that it decreases survival of the animal substantially in the presence of TCS rather than the absence of TCS. They die quicker, you see, time after exposure. You can see that the colon is severely damaged when you have TCS, particularly in the presence of this azoxymethane. And once again here, put on the fancy glasses. Now we're looking at, We're looking at tumor number, increase with triclosan. We're looking at average tumor size, increase with triclosan. And we're looking at total tumor size, also tremendously statistically increased by triclosan in the presence of this carcinogen and this DSS, which is going to cause colitis. Okay, so real huge increase in tumor size because of triclosan. Now we're looking at PCNA, which is proliferation cell nuclear antigen. Okay, and we're looking at beta-catenin, and those are basically tumor antigens. Those are basically tumor antigens. Okay. What you see here in the control, this is just staining for these proteins versus the TCS. Okay. You see that you get an increase in that tumor antigen when you add the triclosan. And here's just histochemical stain, again, showing you the severe damage to the colon. Up here, you're looking at, again, at tumor size, number of tumors, tumor size, and total tumor. Down here, you're looking at how much PCNA staining increased with TCS. You're looking at beta-catenin staining. Remember, that's a tumor antigen, increased also statistically with TCS. Here are all of your innate immune cells. Here's the CD40 leukocytes. You're looking at macrophages. You're looking at the neutrophils. You all got a big increase in this system. Okay. You also get an increase in interleukin 6. You also get other pro-inflammatory cytokines. There's tumor necrosis factor. And here is the actual expression of interleukin 6. And the expression is the transcriptional activity now of the TNF alpha. So it's a transcriptional event in the colon giving you the increases in these proteins. Okay. Again, when you add the additional low dose TCS, it exacerbates colon tumorigenesis using all these markers. There's tumor size, there's tumor number, there's total tumor volume in the liver. All of these parameters, you get an increase in the amount of tumors from triclosan. Okay, so that's what this data is showing you from figure 4. Now, this paper wants to look at the microbiota. Now, I think we all know about the microbiome, right? Microbiome is basically a discussion of all the gut flora, particularly bacteria, although there are some archaea there. So you could ask the question, because triclosan is targeting microbes, particularly bacteria, because it's supposedly targeting the enol reductase from type 2 FAS, which bacteria then utilize for making fatty acids. So that would be the main target, that you might affect microbiota. And what they're showing you here is that diversity of the microbiota is decreased by using triclosan. And that's what this two-dimensional graph is showing you here. You get a decrease in diversity. There's closer to the origin. So the farther from the origin, the more diversity of the bacteria. You see the bigger spread here. You see the differential patterning of the genera of the bacteria that become more tightened as you get less diversification. And here are some of the species. You can run through all these species. So this is again, showing you what increases and what decreases. So this is a heat map. So it increases. Increases are the bright colors, and decreasing are the blue and grays. And you see that there's quite a bit of difference between TCS treatment in terms of bacterial either increasing or decreasing, and that's affected by TCS. So what they point out is that TCS treatment causes 75% reduction of the abundance of a bifidobacterium. Now, bifidobacterium is considered one of the good commensal microbiota, right? It's supposed to protect via anti-inflammation the gut microflora. Okay, so again, that's what they're looking at. They picked it out. Now, when I look at all these different species, of course, they're pseudo species because they're bacteria. I see interesting things. I see decreases in Clostridium, decreases in Lactobacillus. Here's a decrease in some staph, and then here's the increase in strep. So I said, if you go into the molecularity of this, or that is the abundance of specific bacteria, you see lots of things increasing, bacterial strains increasing, but you also see bacteria decreasing, and they're not all protective bacteria that decrease or pathogenic bacteria that increase. So even though they're picking up bifidobacterium and saying, aha, look at this, it's not giving you enough protection. If you really got into this, if microbial ecologists got into this, they would say that there's a lot of variation here. But in general, they're still claiming that you get a decrease in diversity, and maybe that's a bad thing. All right. Now, TCS exposure increased basal inflammation, and it required gut microbiota. Now, this is the important thing. So we saw it was an effect on the gut microbiome. We saw we had an effect on inflammation. We saw that we increased colitis. We showed that we enhanced or exacerbated cancer, colon cancer, right? Now we're saying if we put back in the bacteria and say, are the bacteria necessary for those connections from triclosan to inflammation or triclosan to disease? And it looks like it's so. So with germ-free mice, triclosan has no effect on colon length. With germ-free mice, triclosan has no effect on the expression of interleukin-6 in the colon. Only in the conventional does it, that is, you see, when they have a microbiome, okay? Likewise, interleukin-6 in the plasma, this is the expression in the colon, this is now in circulation. You get what we've seen before. TCS causes an uptick in pro-inflammatory cytokine that you determine in plasma. Significant there at the 0.031 level. That's pretty good. Whereas in germ-free, nope, no difference at all. Okay. Again, here's conventionally raised mice, terrible destruction of the microcrypt. Germ-free mice, well, gee, histochemically, doesn't look like there's any big deal there, shown here on this histogram. It looks like if you don't have a microbiome, triclosan has no effect. So that's now linking triclosan via the microbiome to all the biological, biomedical effects of the drug on the disease and on this particular readout, just looking at whether or not you have colitis. Okay. So control mice, TCS treatment caused enhanced colon length reduction. Enhanced colon length reduction. I like the way they said that. Expression of interleukin-6 in the colon, which is the 6b figure here. That's the transcription. And the concentration of plasma, I just said. However, TCS treatment had no effect on basal inflammation in germ-free mice. And you can see that with the markers for interleukin-6. So that's interesting. All right. Now, results from figure 7. I'm not going to show it to you. It's very simple what it says. Exposure to triclosan activated the TLR4 signaling pathway. And this is in the dextran sodium sulfate induced colitis model, okay, which is what we're looking at here on this paper. Now, what's TLR4? It's a toll-like receptor, isoform 4. I'll show you in a moment specifically what it's involved in. But basically, in general, toll-like receptors mediate innate immunity. And innate immunity readout is looking at things like what? Like circulating leukocytes, and specifically macrophages and neutrophils. Okay? So, toll-like receptors mediate innate immunity via gut microbiota. This is something that's been established in the literature. Using a TLR4 reporter cell line shown there, It was asked whether plasma from the TCS or vehicle-treated dextrose sodium sulfate activated TLR4 actually show an effect. If TCS works through, if triclosan works through the TLR4, so now we're getting down to the molecularity here, because that's the receptor that's involved in the inflammatory response, right? And then mobilization of these innate immune cells, etc., production of cytokines. If TCS works through TLR4, then the connection to the microbiome is at least partially explained via the toll-like receptor-mediated innate immune response. When they asked that question, it appeared so. So that's what that data showed. So to let you get a feeling, a flavor for TLR4, I got this from just from ResearchGate from this particular link on the internet. There's TLR4 binding lipopolysaccharide, which is, of course, a product, an antigen caused from the bacteria, right? It induces this whole cascade system here, right? Through the tracts and through the TRAF, the TAC1. I've discussed this in previous VerEvMed lectures. Through these kinase reactions, there's IC kinase and MK kinase, then interacting with P38, the JUG kinases, the Janus kinases, ultimately causing transcription of all of these particular domains, the Krebs domains, the AP1, the NF-kappa beta domain, and the IRF3. This is going through also just endocytosing of TLR4 or getting double-stranded RNA, which is sometimes associated with viral induced disease in the host. All of these trigger a response that causes survival, proliferation, inflammation, and immune response. That's what happens when you turn on TLR4. So it's a very embedded global pathway which basically turns on inflammation and the proliferation of cells which are involved in that from the innate immune response. Now, one more level of this. This was a paper published in Cell Death and Disease in 2013. And there is actually the site. It's an open access journal, which Nature doesn't normally do, but you can get it there. Darn it. And basically, when you bind LPS, remember that's the bacterial antigen, to the toll-like receptor in a gastric cancer cell now, okay? Remember, this is TLR, is a receptor in the host. It induces reactive oxygen from mitochondria, which causes a phosphorylation of AKT. Again, if you've watched previous lectures, you know what that will do. In this instance, it's turning on NF-kappa B and P65, which then acts as a transcriptional modal, which causes proliferation of these cells. That proliferation is of gastric cancer cells. So obviously, working through TLR4, mediated through reactive oxygen, you're going to get an increase in gastric cancer cell production. So this is not good, right? So anything then that's going to impact TLR4 looks like it's going to cause cancer. And that's what this paper is kind of like tipping its hat to. Back to the data from the paper now. Again, this paper is Science Translational Medicine, published just about a week and a half ago. What this is showing you, here's the activation of TLR4, right? TCS, triclosan increases the activation of it. LPS in the plasma in the presence of TCS, also you see high levels there, right? That means you're getting more LPS. Where do you get more LPS? From fragmentation of the bacteria. Thank you. Gene expression from 16S ribosomal RNA. Why is it doing that? 16S ribosomal RNA, which is a bacterial expression system, increased via TCS. And you see this not just in the blood, but you also see this in the liver. So you're getting a global systemic response, okay, from triclosan in this system, okay? So what is this telling? Now, then they looked at a couple of proteins in part D, and I'll mention that in a moment. But what it's saying here is an increased concentration of plasma bacterial products, okay? And it can be caused by a failure of the intestinal barrier, a function of that, assessed by the effect of TCS on the in vivo intestinal barrier function using fluorescein isothiocyanate, and that's shown down here. So when you get an increase in that, okay, in the plasma of that particular fluorescein marker, it shows that you're getting the degradation of the intestinal barrier, okay, because it's coming out into the plasma. That's what that means. It's being secreted because you're getting a perforation of the intestine. So you can get an increased intestinal permeability by finding FITC dextran in the plasma. And you see that triclosan enhances that. Now we look at the expression of colonic proteins, which are normally considered critical for maintaining the intestinal integrity of permeability. And those include proteins like occludin, ZO1, mucin 3, and TFF3. Those are proteins that are considered protective. Trefoil factor 3 is what, and the other one is this tight junction protein 1 or ZO1. It revealed that only looking at the expression of these proteins, okay, only the expression of occludin was reduced. So the other two seem to be functioning okay, no downturn in the expression of those proteins or accumulation of them. But occludin does seem to be greatly reduced by triclosan. So that could suggest that triclosan induces the increased intestinal permeability by down-regulating the level of occludin, which would normally maintain the integrity of the intestine. So that's what that's saying there. All right. Figure 8, we're almost done, don't worry. TCS, triclosan, enhanced DSS-induced colitis through a TLR4-dependent mechanism. So now we're making sure that all this works through TLR4. Now, and the way that they're doing that is how? They have a TLR4 double knockout. Okay. Now notice, first of all, look at the straight up plot here, looking at body weight. Notice that body weight is best in wild-type mice control, but when you get rid of the TLR4, okay, shown here, right, the body weight is even more reduced. That's what this should show. Well, excuse me, the body weight is maintained when you get rid of TLR4. When you have TLR4, okay, when you have it, that's here, this wild type, you get a larger decrease in body weight. That means that the triclosan, in the presence of the stimulant of colitis, is causing a faster decrease in body weight when you have the intact TLR4 receptor. That's what it's telling you. So again, when you knock it out, look at how you get an increase in body weight as compared to when you have wild type. You see that there? That's because you're no longer functioning through the TLR4, which suggests TLR4 is at least partially responsible for the triclosan response. That's exactly what it's showing you here. Okay, you get a better colon when you get rid of the TLR4. Right, you get less damage than you do in the wild type. That's what it's saying. This is colon length here. You see, when you have wild type mice, it's reduced from triclosan, but when you knock out the TLR4, the toll-like receptor 4, which again they believe is the mechanism by which triclosan is functioning here by turning on that entire innate immune pathway, you don't seem to get a decrease in colon length, which is their readout here. Likewise, we're looking at here again at innate immune cells. Here's total CD45 leukocytes in the colon. You remember we saw an increase in the wild type. You get rid of TLR4, you do the knockout of that gene, right? Hey, no effect on circulating leukocytes. No effect on circulating neutrophils either, okay? So look at that, there's just no effect. All right, likewise, if you look at interleukin 6, the pro-inflammatory cytokine, you get rid of TLR4, right? You get rid of the toll-like receptor, which I believe is the mechanism by which this mediation occurs. Doesn't seem to have any effect. You get no increase in interleukin 6 by using triclosan. See that? No increase compared to control. Whereas you get that whopping amount we saw before. So it looks like it's a TLR4 mediated event. Now, let's do a discussion of what all that data showed us, shall we? Off with the glasses. Short time exposure, just a couple of weeks, to low dose TCS, the kind you would have in human exposure, for example, caused a low grade colonic inflammation. They got increased colitis when they induced it with DSS, and they got a colitis associated colon cancer in mice when they added the azomethane compound. All of that doesn't look good. It looks like triclosan is exacerbating this problem substantially. So it might suggest there's a potential adverse effect of triclosan on human gut health. This is a mouse model, not human. So they're saying maybe you could extrapolate this to why we're getting an increase in IBD, and then associated with an increase in IBD, an increase in colonic cancer. That's potentially what they're saying here because of triclosan in the environment. And they say, or they showed, that after mice were chronically exposed to either 10 to 80 parts per million TCS in the diet, which is how you would get TCS, the concentrations in the mouse plasma were comparable or within several folds of concentrations reported in what you find in normal, healthy human volunteers, right, in the urine. Comparable plasma levels of TCS in the exposed human population therefore might alert the medical community to potential environmental risks. Okay, so this might be something to look at. This is what they're trying to argue. They haven't demonstrated this, they're just arguing it. Now, their work seemed to correlate intact microbiome. Remember, you needed that with the germ-free mice, you saw no effect. Intact microbiome associated pattern recognition membrane receptor, that's the TLR4 component, mediating the innate immune inflammatory response. Remember the interleukin-6 and tumor necrosis factor alpha expression in the colon, and also indicative as shown in the plasma. And they're saying all of that is a potential mechanism linking the triclosan to colitis or inflammatory bowel disease in general, and maybe even colon cancer. That's what they're trying to say, and that's how this paper became published just recently. Now, there are potential mitigating biochemical cellular interactions with triclosan that might or might not correlate with what this paper seems to have delivered us. That is human IBD and colorectal carcinoma. Okay? Now, consider the periodontium. Remember that triclosan is used in toothpaste. And one of the reasons it's used in toothpaste is supposed to help control or modulate plaque formation in the oral cavity, and in so doing, decrease the production of cavities. Okay? That's why it's in the toothpaste you use. Okay? Now, inflammatory periodontal disease can result in soft and hard tissue destruction. So you can get quite an inflammatory response in the mouth. And when that happens, then you have really severe dentition, and you can have real problems in overall dental health, right? Oral health. The microbial factors that are involved in that disease state are only the etiological agents, but they don't really cause the disease. That is, they're not toxins per se. Rather, they induce the inflammatory response, and that's where you get the tissue damage, right? In the periodontium. Now, cells of the periodontium, including the epithelial cells, all important, fibroblasts and monocytes as well, and they all express TLRs, okay? And with oral epithelial cells acting in the first line of defense, because the other cells, obviously, fibroblasts are generated later, right? Because they're not actual authentic epithelia. And of course, the monocytes are induced or mobilized because of inflammation. So epithelial cells actually serve multiple immune roles, along with the mucosal surface functioning as a physical barrier. That's what the epithelial cells are. A source of antimicrobial peptides. In case you didn't know that, they sure do. Inflammatory immune cells themselves, that's how they act, which produce inflammatory cytokines. And for these tremendously important epithelial cells are also involved in tolerogenic immune association, right? Producing things like interleukin-10. And that can produce these immunoregulatory cytokines like IL-10, or they can remain quiescent and therefore not contribute to the inflammatory response. So these mucosal epithelial cells are really important for normal oral health. Triclosan decreases gingival, right, inflammation, and has a direct antimicrobial effect, as we already know. That's what it's touted to do, inhibit that enol reductase. And one of the mechanisms, though, that they pulled out here is, interestingly enough, through the toll-like receptor. So a toll-like receptor induced inhibition. This has nothing to do now with fatty acid synthesis. Check this out. Directly targeting TLR. A TLR-induced inhibition of cyclooxygenase and lipoxygenase pathways. Those are going to produce pro-inflammatory eicosanoids, which will then trigger an innate immune response. So this is fatty acid metabolism, but not synthesis. This is oxygenation of fatty acids, particularly C20 fatty acids, particularly the omega-6 series, so that you generate the inflammatory response. Remember that NSAIDs function against those pathways, particularly cyclooxygenase, that is. So that results in decreased prostaglandin E2, leukotrienes, and interferon gamma, all of which will promote inflammation. via initially the cyclooxygenase and lipoxygenase or COX and LOX induction of the system. And triclosan decreases all of that, right? So triclosan mediates an anti-inflammatory effect on, neutrophils, we just talked about those, monocytes, we just talked about those, and lymphocytes, okay? So all over the map are we affecting then the immune response. Now we're talking about lymphocytes, T and B cells. Okay, so take a look at this. Okay, this is this oral disease paper. Here are monocytes, okay? So as you increase the amount of triclosan, the amount of interleukin 8 decreases. Interleukin 1 alpha decreases with increasing triclosan. The amount of tumor necrosis factor alpha decreases with triclosan in monocyte macrophages. How about in human keratinocytes? Well, now these are epithelial cells, right? No effect on interleukin A, no effect on interleukin 1 alpha, no effect on tumor necrosis factor alpha. Okay, so these don't seem to be reacting the same way. Let's go back to another cell type, the fibroblasts, the third one they looked at. Yeah, increased triclosan, decreased all these are pro-inflammatory cytokines. Okay. And the optical density doesn't seem to be affected by triclosan. You're not inhibiting growth. If you're inhibiting growth, you'd get less of the protein product, and that's not what you're getting here at all, right, with these three cell types. That HOC is actually the keratinocyte, which is the epithelial cell from the mucous membrane. So triclosan decreased pro-inflammatory cytokine secretion, and they stimulated this, of course, with LPS. So you're going through the TLR4, remember. In monocyte and fibroblast primary human cell lines, these are taking cells now, putting them in culture, okay? Nothing to do with the microbiome. Microbiome, totally not a player here. You're looking at cells in culture, and then you're inducing with LPS. So you're working through the TLR receptor, TLR4, and then you're adding triclosan, and you're looking at whether or not you get an inflammatory response, and you get it diminished in monocytes and fibroblasts, but not in keratinocytes, at least in this experiment. And of course, it had no effect on cell viability. Now, pre-treatment with triclosan prior to the LPS stimulation. So pre-treatment, this could be like, for example, triclosan in the environment, right? Blunted the inflammatory response. Blunted, didn't activate it. In the epithelial HOK, those are the keratinocyte cell lines. But it increased defensin secretion, which is, of course, a good thing. So here's the data. There's interleukin 8. You see here that when you add LPS, you get a lot of it. Here's unstimulated versus stimulated. Here, when you have triclosan, you do this stimulation, okay? LPS, it doesn't seem to have any real effect, right? No real big effect on interleukin 8. No real big effect on interleukin 1 alpha. No real effect on tumor necrosis factor alpha. So what you're seeing here is that LPS does not seem to function if you pre-treat with triclosan. Remember, before we looked at those keratinocytes, those primary epithelial cells, and it didn't seem to have any effect. But when you pre-treat with triclosan, it blunts the whole system. Now, these are defensins, and you see an increase in defensin production in the presence of triclosan. It's the beta D2 and beta D3 defensins. So triclosan may interact. Now, what's going on here, right? This isn't the microbiota. This isn't affecting bacterial enol reductase for sure. These are cells in culture. So what they're saying is that triclosan may interact with membrane lipids directly, thus providing a protective mechanism. Now, why do they say this? Because that's in the literature. Why haven't I emphasized in this talk? Because it's a huge literature, okay? Just talking about the physical chemistry of the membrane interacting with triclosan. Nothing to do with the enol reductase, nothing to do directly with the immune response. Only membrane alteration. Membrane biophysics, biochemistry is affected by triclosan. I told you triclosan, just like all compounds you see out there in the pharmacopoeia, have pluripotent effects, right? There's a plenary of effects of triclosan on biological systems, not just on one enzymatic reaction or even on a whole global pathway like innate immune induction. Now, defensins, which is again shown down here, the increase in defenses because of triclosan, see, in the presence of LPS, they are actually what the body does to produce, to inhibit the bacteria. Now, these are peptides, okay? So defensins are antimicrobial peptides, generally considered as protective of the oral cavity, via either suppression or selective activation. Yeah, they work through the inflammatory response via differential TLR signaling because there's multiple toll-like receptor system. Expression of the LPS receptor, or TLR4, that specific one that binds LPS, or expression of the intracellular signaling protein downstream, remember all those proteins we showed you in the previous slide, of TLR4 can be altered to enhance or lessen the reactivity of LPS, therefore decreasing inflammatory response. So this is quite different than what we just saw with that whole colon paper, right? Yeah, totally different. This is suppressing the inflammatory response in the oral cavity. So let's discuss this oral disease 2013 manuscript relative to what we just did with the colon manuscript. Triclosan improves clinical plaque control and slows periodontal disease progression. That's why it's in your toothpaste. Triclosan does have, of course, an antibacterial activity as well, right? Because of the inhibition of the enol reductase. Plus, the anti-inflammatory properties via the TLR signaling that we just talked about here, affected by triclosan, provide an additional benefit in the management of the inflammatory periodontal disease paradigm. Monocyte or fibroblast activation was perturbed by co-treatment with triclosan and LPS, whereas epithelial cells required pre-treatment with triclosan to prevent inflammation. So that means that the induction period, you have to, in other words, poise these epithelial cells with triclosan for them to blunt the inflammatory response. And why do you get that? You get that by having triclosan prophylactically in your toothpaste or in the environment in general. All right. So triclosan, that diphenyl ether that looked a little scary to some people probably, actually promotes accelerated lipopolysaccharide tolerance. Remember, that's going to be a product of bacterial degeneration. It's going to be acting as an antigen to induce the innate immune response. So it's going to accelerate tolerance to LPS through the regulation of the toll-like receptor-induced signaling, thus preventing overactivation of the keratinocytes, perhaps through membrane lipid, as they waved their hand and suggested here. But they actually looked at miR-146A, right? That's microRNA, and that microRNA seemed to be elevated by triclosan. So now we're looking at an epigenetic event. Remember that microRNAs will bind to messenger RNAs and prevent translation and therefore act epigenetically. So this interfering RNA, 146A, very common in the literature. It seems like you get an uptick of that in the keratinocytes via the TLR receptor, via interactions of that receptor, and that blunts the inflammatory response via downstream degradation of expression of the proteins necessary for the full-blown, fully florid TLR pro-inflammatory response. So now you are invoking an epigenetic phenomenon, starting to look a little bit like a dieventological event, right? Of course. So here's another paper. This is Toxicology in Vitro. It's the name of the journal, published just a year ago. Year and a half ago. Triclosan can promote, now check this out, another disease system, hepatocellular carcinoma in another mouse model. Triclosan can induce global, here we go, full-blown epigenetic effect. Triclosan seems to induce global DNA hypomethylation in these particular hepatic cells, these HepG2 cells, right? These are human hepatic cells. Now, hypomethylation means less methyl groups. We've talked a lot about the epigenetic marker of methyl groups on cytosines in DNA and how that generally suppresses expression of genes that are methylated. Here we're causing a hypomethylation, which means that rather than getting a decrease in the activity of the expression of those genes, you might get an increase in whatever genes are being affected at the plenary. Now, after exposing these hepatocytes to low-dose TCS, look at how low that is, 0.625 to 5 nanomolar, very low, for just 2 weeks. Of course, it's cell culture, so you're going to have lower concentrations necessary for an event. There was a decreased repetitive element line 1 methylation level. Now, line 1 are just repetitive elements in the genome. They're actually used as markers to study evolution. I know that because my daughter used to work in a lab where they were looking at hypo and hypermethylation of line 1 elements when she was an undergraduate. That's what she did. But not global DNA methylation. So only those line 1 elements. So that's where you get the hypermethylation at. Through down-regulating, now look at the way it works. Down-regulating the methyltransferase, the DNA methyltransferase 1, that's the DNMT1, and the MECP2, which is the methylated DNA binding domain. That protein protects the methylome, prevents the methyl group from being removed. So it down-regulates the methyltransferase, and it down-regulates the MECP2. What that serves to do in general is cause hypomethylation. Now, here's the real interesting missile into the complex, right? Following low dose TCS, elevated hepatic tumor suppressor gene P16 methylation, okay, thus blunting P16 expression that was not observed in high dose TCS, okay? Low dose elevated methylation on that particular gene. Now, why is it causing hypomethylation on line elements, which we just told you, but it's actually increasing methylation of P16? Well, because it's a differential regulation, right? This is a cell type. It's a living system. So living systems do not work monochromatically. They don't all go in the same direction. They're not all followers. So when you're decreasing the methylation of generalized areas of the genome, you might actually increase the methylation of genes which otherwise cause more specific effects. And when you methylate the hepatic tumor suppressor gene P16, what do you think happens? Expression of that gene drops. What does that do then? Enhances the hepatocellular carcinoma. That may have been the real reason all this methylation is going on. The hypomethylation might just be like an indicator of what's going on, or maybe even a very poor biomarker. The real thing is the hypermethylation of P16, thus inhibiting tumor suppression, thus increasing hepatocellular carcinoma, all because of triclosan. So a derivative, they went on to, they were doing a little bit of pharmacology here, a derivative methyl triclosan, they added a methyl group, that hydroxyl, totally ineffective. So you need the hydroxyl group. Not surprising, hydroxyl is a highly reactive chemical moiety, so that doesn't surprise me at all. But this maybe suggests not only interacting directly with the TLR, but also with the membrane. Okay? Free hydroxyl is going to react quite extensively to polar groups in the membrane, like in polar lipids. Essentially, like ethanolamine. Essentially, or serine, ethylamine, phosphatidylserine. Sorry for the aside. Essentially, this paper showed low concentrations of TCS, that's the triclosan we've been talking about, adversely affect hepatic cells through DNA methylation dysregulation. Hypomethylation one place, hypermethylation another location of the genome. And the hydroxyl group in triclosan seems to be playing the dominant pharmacological role here in the effects thus associated with the antibiotic with epigenetic mediated, this is all epigenetic now, hepatotoxicity. Now, let's do a diaventomic analysis of this triclosan-mediated biomedical consequence, just from the few papers I've assembled, collected, and discussed right here on the 9th of June, 2018. Biological adaptation to a purified antibiotic like triclosan involves the host and the microbiome. Okay, we can put that out there as a premise. Appears to be the case. Host cells, including general leukocytes, lymphocytes, epithelia, keratinocytes, respond differentially to triclosan. Differentially, key point there. Directly and indirectly as mediated by resident flora. So directly via things like TLR4, and then indirectly membrane-associated alterations, right? Those are two possibilities right there. Both elevated and suppressed inflammatory responses were noted today. And these could be tissue and resident organ specified and valence. Valence meaning, okay, which tissue you're talking about, which organ you're talking about, and whether or not it's up or it's down. So that's the valency of it. That there is a correlation of triclosan with inflammatory bowel disease and potentiating not only colorectal cancer, but hepatocellular carcinoma, which is also linked to IBD, by the way. So the literature suggests you get colorectal cancer, but also you get hepatocellular carcinoma via inflammatory bowel disease. That is not direct, even a direct correlation. It's certainly not a causation, but there are links. Suggests as being linked to a pro-inflammatory response. That looks right. But it's possible that there are epigenetic reprogramming signatures, which we just saw, that may alter this dysbiotic descent into disease. Okay. Studying then specific cell types, say lymphocytes and leukocytes and hepatocytes and colonocytes and epithelial cells in the mucous membrane versus fibrotic cells versus monocytes versus all the innate system cells we're looking at. Studying specific cell types, host tissues, and all the resident flora, remember the microbiota diversity slide, both pathogenic and commensal, that is the flora there, and the event interactions, so real-time events, not just the substances, no matter here. Remember, no matter, it's the event. What was a good indicator of that? Remember when you pre-treated with Triclosan? That was an event induction. When was it there? When, you always have to think of a time course. Time course always is event ontology. All right. With genotype and epigenotype, so we know nothing really about what's going on with the genotype of these organisms. These are all isolated research papers, not spending much time on, although we did look at a knockout of IL-10. We did look at a knockout of TLR4, so we're starting to look at genetics there. The epigenotype, well, hypermethylation, IL-16, all that stuff. Or IL-8, I mean, and the P16 suppressor, tumor suppressor protein. Okay, those would be epigenotypes, plus the MER 146A, that was another thing, right? The interfering RNA species, microRNA species. All of that epigenotype in concert with the immune response, the general immune response, like pro-inflammatory cytokine production or anti-inflammatory cytokine production, helps explain, it's all it's doing is explaining the cross-reacting eventual interactions that ultimately leads to this biomedical consequence. And those could be anything from colitis to not colitis, from protection against gingivitis to alteration of the plaque bacteria that enhances periodontal disease, something you also find in the literature. And of course, flat out antibiotic resistance inducing bacteria that are no longer even susceptible to triclosan. All of those things are under the umbrella of the diaventome. Okay? So that's a really important consideration to have in mind, that when you look at an isolated paper like the first one, looks like, wow, Triclosan really, sort of a bad player here. Then you look at the periodontal disease paper, it's a good player. Then you look at the hepatocellular carcinoma, back to bad player. So you cannot simply say that Triclosan in the environment or in the diet or in your toothpaste is going to be a direct negative consequence. One thing for sure, though, constant selection pressure against the microbiota, you are going to make them resistant to it, if that matters. Okay, so that's the end of today's talk. This picture was taken over the Clearwater River around 2:30 in the morning with a waning moon. It's a really beautiful sight. The picture is not nearly as good as what it looked like when I was walking up there. That's where I walk every morning. All right, that's me, Dr. Daniel J. Guerra. That is my email address. Please contact me. I'd love to talk about this talk or any of the other seminars I've given, and of course about VerEvMed. There is the Facebook page. Please go to it. You're going to find this video lecture as a YouTube lecture embedded there soon. And there, of course, is our website. And that is our email address to our website. So please contact us. We'd love to discuss things with you. All right. So I'm not going to go any further than just to say goodbye for now. And we will see you real soon. So where possible, always have an authentic day. Bye for now.